over to your talk.
All right, thanks, Luke. Thanks for your very nice introduction. And also, of course, I would like to apologize for today's delay. My computer was crashed, so I have to run to the lab and use a different one. But yeah, for today, because I actually lost my previous slides, so what I'm going to talk about will be slightly different than what I plan to do. And so here I'm going to also, in addition to the dynamo work I'm going to present, I will also talk about the spiritual work that I'm currently working on. All right, without further ado, let's jump into the actual talk. So I'm for many years, I'm always interested in moving towards achieving predictive modeling in biology. So in many ways, this is similar to the discovery of a fundamental principle in astronomy that happened almost 400 years ago. As we know, astronomers like to understand how the planet moves. And 400 years ago, Tycho, thanks to the development of the telescope, he made comprehensive observations of the planetary movement. And then this big data was carefully analyzed by Kepler to derive 3 empirical laws of planetary movement, which eventually led to the discovery of the theory of gravitation by Newton. Now, in biology, we have many big data sets. And one of the questions I always wondered is that can we also obtain uniform theories or quantitative models in biology with this big data? Similar to astronomy, we as a biologist would like to understand how does the single photolized cell give rise to an entire organism, our time and space in a precisely controlled manner. And I have been thinking about this problem for many years, especially I was deeply inspired by this metaphor of epigenetic landscape by Conrad Washington that was proposed almost 60 years ago. And during my master's degree, almost 10 years ago, I actually worked on applying some mathematical models that map the epigenetic landscape of the canonical toggle switch network. So I started with writing down the differential equation of the system, and then I can mathematically quantify the epigenetic landscape, which then leads to quantification of the optimal developmental de-differentiation and trans-differentiation path based on the principles like the destruction path in the physics. Of course, all this work has been done in theory, and and at that time, one of my biggest question I asked is that can we able to integrate my theoretical model with the genomics data that just started 10 years ago. And this is exactly what I delved into in the next 10 years or so in my PhD and postdoc. So in my PhD, as Luke mentioned, developed molecular 2 and 3 for the pseudo-time ordering, working with a core, and then also developed the script that enable us to infer the causal gene regulatory network. And in my postdoc, and worked with Jonathan and others to develop a new tool for modeling the single cell genomics data, the dynamo framework, which I'm going to also talk about in detail today. On the other hand, my master's also contributed to analyzing many breakthrough technological developments, including like scale RNA-seq, ATAC-seq, and the SCNT-seq, and recently the stereo-seq. Going back to this problem of embryogenesis, now thanks to the emergence of single-cell genomics approach, we are able to generate really many unprecedented profiling of the embryogenesis at a very high scale. that enable us to understand this cell state transitions and also developmental trajectories. But conventional tools for analyzing the single cell data are relatively descriptive. Most of them will only try to identify the cell clusters that corresponding to a particular cell type and also the associated marker genes. And using tools like Monaco 2, Monaco 3, and others that we and others developed, we can also try to infer the developmental trajectory by pseudo-timing, by ordering the cell along an underlying graph manifold. Alternatively, people also come up with the idea of ion velocity that essentially leverage the physical kinetics of the RNA RNA life, and then we can derive a measure of the local dynamics of each individual cell, and then we can project this velocity to lower dimension to reveal the cell state, the feature of the cell state in a shorter time period. So all these tools, although are very powerful, they still have a lot of fundamental limitations. For example, with the pseudo-time, you can only able to reveal the central trend of each of the cell population, and most of the time, the tools people developed cannot tell you the routes, and we have to manually set the route, the beginning state of the pseudo-time trajectory. On the other hand, ion velocity make a lot of undesirable assumptions. For example, assume the transcription rate is a piecewise constant, the splicing rate is like universally constant, and the data that they used for ion velocity estimation previously relies on the introns, so it's very which is really biased and also unreliable. Almost 4 years ago, when I started my postdoc in Jonathan's lab, I wondered, can we actually go beyond this more descriptive analysis of the single cell data to a more predictive modeling? And to me, this we will need two advances in two different areas. On the one hand, we will need a more sophisticated model that goes beyond to the descriptive analysis, like the discrete ion velocity vectors of the single cell, to a continuous and differentiable function of the entire system. And then it obvious to me that once we have such an equation, such a model, then it will open doors to many fundamentally different analyses that are not possible with pseudo-time and also ion velocity approaches. For example, with this continuous equation, we can use it to predict the cell fate. It's shown in this animation. We can also use it to resolve the regulatory network, and even predict the optimal reprogramming paths and the transparent factors, and even making non-trivial in silico perturbation predictions. On the other hand, I also realized that we will also need to have new technologies that enable us to get better measurements for ion velocity quantification. And for this, we use the metabolic labeling enabled single cell RNA-seq. Working with Xiaojie and Chen from Vijay Sankar and Eric Nender's lab, we performed a single cell sequence experiment of the human hematopoiesis. And then we try to use the splicing data from this experiment to prove ion velocity. As you can see, the ion velocity resulted from the splicing data is very problematic and also noisy. For example, we see like megakaryocytes, erythroid, basophil lineage, they actually move backward to the progeny. Well, if we use the labeling data, it automatically smooths out the the iron velocity flow and reveal a very nice hierarchical transition from the HSC firstly into like GMP, MEP lineage, which then ramifies into neutral field, monocyte, basophil, erythroid, and megakaryocyte cells. Just nicely recapitulate the known biology of the human hematopoiesis. So this establishes the fact that using labeling data is in general will improve the iron velocity measurements comparing to the splicing-based data. And then we developed a comprehensive tool that tries to build an end-to-end framework for analyzing and modeling this kind of data. So Dynamo actually has four major components. Firstly, it will develop a comprehensive estimation framework that deals with both the conventional and also the labeling single-cell iron sequence data to estimate the canonical parameters that includes like transcription rate, splicing, and degradation rates. And with this improved iron velocity rates, which is also absolute, meaning it actually have like the physical units, and then we can get the absolute iron velocity. And then with the improved iron velocity, we can then try to learn a continuous and differentiable function in the transcriptomic space, which we can then leverage the differentiable property of this system to calculate the height of the differentials. So we can go beyond the iron velocity to like iron acceleration, coefficient, divergence, Jacobian core, Hessian. And we can then use this just each of this property to make a game mechanistic insights. And lastly, making non-trivial predictions using distraction paths on in silico perturbation predictions. So at the core of Dynamo, it's just its ability to tag the paired measurements of the current gene expression X and also the associated velocity vector dx over dt for many cells, and then we use a machine learning algorithm to essentially learn a continuous function that map the gene expression X to the velocity dx over dt. And then this function fundamentally relates to the underlying gene regulatory network, which in general is a very high dimension, and we don't really know the exact form of this equation. But once we have such a function that learn through data-driven approach, we are able to use it to make a prediction such as the prediction of the individual trajectories for each cell. So without going too much into the technical detail, this approach, this vector field is represented as a summation of a series of basis function, where the x and tilde is corresponding to a landmark, and this tau is it's like a Gaussian kernel, and the Cn is the weight for corresponding to each base function. And since we have an analytical form of this vector field function, so we can actually use it to make sense of this vector field. So what I'm going to tell you next are some computational some concepts that you probably learned in your calculus class, but they haven't thought of for a long time. All that matters is that all these terms will actually have some deep biological meanings, as I will explain. So firstly, we have the iron Jacobian, which essentially tells you the changes of a changes of the velocity of a particular gene when you change the expression of another gene. So what this means is that if you give me a single cell data, I can actually estimate a gene regulatory network for each individual cell. Well, for the divergence, it defines as a trace of this Jacobian matrix. So this will help us to reveal the source and sink of the cell state. And with acceleration, which is the derivative of the velocity, it can help us to reveal the earlier drivers And with the curvature, we can start to talk about the geometry in our data, which we know sometimes are very difficult to quantify in the gene experience space. And then with the core, it potentially relates to the cell cycle processes. So in the next few slides, I'm gonna give a few examples to highlight a few terms that we can use to gain biological insights. So in the case of divergence, as I mentioned, it defines as a trace of basically the sum of the diagonal of the Jacobi matrix. So we can calculate essentially a value for each individual cell. And if this divergence is negative, it indicates the local flow is convergent. So this may indicate that we have a sink. On the other hand, if the divergence is positive, it will indicate that the local flow is divergent, is going out. So it may indicate a source, which may also correspond to the progenitor state. In fact, if we apply this approach on a dataset we previously generated of the zebrafish pigmentation, we found that the middle progenitor cells, like Schwann cell progenitor, pigment cell progenitor, they all have a strong positive divergence, where the surrounding cells, just terminal cell types, like iridophore, melanophore cells, they actually have a negative divergence. This indicates that using divergence, we are able to automatically distinguish the progenitor and also terminal cell types, essentially for any systems. Another fascinating property is this RNA acceleration, which can tell us the early driver. So let's look at this particular case of the acceleration of the SPA1 and the GALA1. These are two master regulators of the human hematopoiesis. And we can look at the transition from HSC to the monocyte lineage. And then we can leverage the vector field that we calculate to compute the acceleration, velocity, and expression for these three genes. Interestingly, you can see the gene SPA1 or PU1 actually has the acceleration of this gene actually peaked at around 21 hours, while the velocity of this gene peaked at 47 hours. And then the expression of this gene peaked at a much later tempo, like 70 hours. This indicates that using the acceleration, we are able to predict what is going to happen in the future. So in principle, we can use this approach to predict the early driver of the cell fate transitions. I have told you that using the differential geometry approach, we can able to gain many mechanistic insights. So for example, we can predict a minimal network that explains the megakaryocyte lineage, the early appearance of the megakaryocyte lineage. On the other hand, since we now actually have an analytical function, we can use it to also make many non-trivial predictions. For example, we can use it to predict the most probable path, which is essentially defined as the trajectory, the difference of the trajectory respect to the vector field along a path. And then we can also we can also predict the perturbation effects after locking out the gene. So in the next few slides, I'm gonna get you through how we can use the recession path and also the cyclical perturbation to gain predictive insights. As you know, previously on this simulation system, unable to use the recession path method to predict the optimal developmental division and the transdivision path. And now, since we learned the vector field on the real data, ideally we should also able to apply this approach on the vector field we learned from the data. And what I shown here on the bottom left is the predicted developmental path we learned for the developmental lab we predicted for this system. So if you look carefully, you will realize that, interestingly, the lab is not the shortest path that connects the HSC to each of the terminal cell type. Instead, it follows the vector field, follows the string name defined by the vector field. This is because if you look at the definition of this list action, this action is here. If the trajectory corresponding to the vector field, then the first term in the integral will correspond to 0, and that's way we will get the list action. Another thing I want to highlight is that although our list action path shown here is on the two-dimensional space, we can actually project this list action path back to the original gene expression space to reveal the transcriptomics kinetics along the lab. And what I'm showing here is the gene expression changes from the HSC to the basal field along this lab path. So you will see some of the gene will turn on much earlier during this development process, where other genes are much later. Then we can then we developed another method that tries to calculate the accumulative changes of each gene along the path, and then we are able to prioritize some important regulators for a particular developmental transdivision events. And in this case, we find that the GALA2 RANX1, just a previously known master regulators of the basal lineage, are indeed ranked way ahead with this score. So we then try to apply these methods to make more global predictions of the hematopoietic cell fate transitions. In this particular case, we are looking at the top, the predicted essentially the priority score for each transcriptional factors for the transdifferentiation events for the hematopoietic system. So you can see most of the transcriptional factors are actually receive a priority score above 0.8, with 1 as the perfect score. And if we use if we perform a global analysis of all the known transitions, and we found the AUC, the area under the curve of the RAG curve analysis reveal a score of 0.85. So this really indicates the high accuracy of this approach. Now, since we established the accuracy of this approach, we can then apply these methods to the entire cell-fate transition to obtain a transition matrix for the entire hematopoietic system. The transition essentially from any hematopoietic cell type to any other hematopoietic cell. And there will be an interesting future direction to validate these predictions for just yellow boxes, and those are the ones that are previously not reported. In order to also highlight the predictive power of our approach, we next designed another in silico perturbation methods. And again, here we're gonna use this PU1 GALA1 network as an example. So as you can see, because of PU1 GALA1, they have these two genes have self-activation and also mutual inhibition. So it behaves like a seesaw model. And if we suppress the expression of GALA1, then obviously this balance will be broken, and then the SPA1 should win, and then the cell should, in principle, will move towards the GMP lineage. And then using our in silico perturbation methods, which essentially just calculate the dot product between the Jacobian matrix to the data X. Data X is the essentially the change of the gene expression for a particular gene of multiple genes. And then we can apply this approach to make the simulations of this GALA1 knockout. Interestingly, as you can see, if we suppress the GALA1, indeed the cells from the erythroid MAP lineage will move away and move towards to become the monocyte neutrophil, consistent to our previous explanations. And this, of course, is also validated, also consistent with the previous biochemistry experiments. It's also worth noting that our approach is actually very efficient, and it can be genome-wide and also combinatorial. So this will, in principle, provide a complementary methods for just genome-wide proteomic efforts, like what we have done in the Weizmann lab. So recently, we realized the fact that the clonal method in general is less powerful comparing to the modern deep learning approaches. We also developed a new approach that improves the clonal method from Dynamo, and we designed a Dynamo framework. Dynamo not only able to learn the vector field, but also extend this framework to other modalities. So we can use it to learn the vector field from, for example, like the population 10 series. We can also learn vector field from proteomic data, and even if we have this trajectory data, we can also use it to learn the vector field from the trajectory data. So for the sake of the time, here I'm not going to tell you the exact details, but in general, for the velocity data, we basically use autoencoder plus like a fully connected neural net in the middle to learn the vector field for the velocity data. And then for the trajectory and population data, we use NeuOD. For population data, we of course use the optimal transport. And we found our results when using this deep learning approach dramatically improves the Dynamo approach. So as you can see previously with the Dynamo, the predicted Jacobian has a lot of noise, especially in just boundary regions. When we use the Dynamo framework, we are able to dramatically improve this estimation and achieve nearly perfect recovery of the Jacobian. Now, I have told you that using Dynamo, we are able to achieve the modeling on the temporal axis. But as I mentioned at the very beginning of my talk, the embryogenesis is not only temporally but also spatially controlled. So in order to achieve the spatial modeling, similar to what I have done for the temporal modeling, I also needed to identify the ATL technologies that achieve the best data for the best modeling. For this, and collaborated with BGI research previously and utilize their stable secret data to achieve the spatial modeling. The existing technology for spatial spatial profiling are in general very limited, because they use just most of these methods like techniques of vision and also slide-seq use beads, where each bead corresponding to multiple cells, and also the distance between each bead is very far. So this method is not able to achieve the single cell resolution. We are using stereo-seq. We actually use we leverage the sequencing chip, and so this method will give us very heavy resolution, because on the chip we have DNA nanobots, and then we can obtain up to 400 DNA nanobots per individual cell. On the other hand, we also have large field of view, so we can easily capture an entire mouse embryo with a single chip. And in principle, we are able to capture up to 13 centimeter by 13 centimeter field of view. So in principle, we are able to profit. We can profit any human organ at its entirety. So this plot essentially demonstrates that comparing to existing approach, we really the stereo seq really improves the spatial resolution and also field of view by several magnitudes. And in our previous study, we try to with the showcase the power of this method. So we profit a Morse embryo at E16.5, and then we perform the single cell segmentation of this data set. And once we have the single cell segmentation, we can just treat these cells as a conventional single cell RNA-seq did to perform like a normalization, transformation, and also UMAP, as well as clustering. And what I shown on the left is the identity of the cell type we from this data set, and then we can project these cells back to the original physical space to reveal the intricate spatial distribution of each cell type. Because this spatial data actually has very high RNA capture, so we can also able to perform spatially and temporally resolved RNA velocity analysis. And here I'm going to highlight this analysis for studying the neurogenesis. As you can see, this analysis reveals two orthogonal RNA velocity process. So on the bottom, we see basically a tangential migration, which is characterized with different genes that previously shown to be related to this migration. On the top, we also reveal like a radial migration, and then this again it's characterized with the expression kinetics of like PEG6, SOX2, just also known transparent factors for this radial migration. And I think this because as I said in the beginning, I'm using the slides that are not really what I wanted to talk initially. So maybe I should switch to a different one now. But before I switch, if you have any questions, please let me know, and I would love to address them now. Okay, then maybe I should, because I also only have 5 minutes, maybe I should quickly go through the other slides I wanted to talk. Yeah, so as I mentioned, that we now with the stereo seq we are able to achieve very high resolution, and we large field of view of spatial transcriptomics. So this kind of data offer us a new opportunity to develop a powerful tools for studying cell-phase transition at a single cell resolution and 3D level. So we recently also developed just a new tool called the Spatial, which enable us to perform single cell segmentation, spatial domain identification, negative receptor interaction, and the 3D reconstruction. So for the sake of the time, I'm gonna quickly tell you that now with Spatial, we are able to achieve precise single cell segmentation, and we are actually implementing a new tool called the Staro. This is actually part of the Spatial framework. Without going too much in the technical detail, so the Staro will basically tag the gene expression matrix with the spatial coordinates, and then leverage a EM algorithm that models the front and the background signal of the RNA to identify the front and also the background RNA signals, which then augmented with the brief propagation approach to achieve the single cell segmentation. we also have alternative ways that are based on Gaussian blur and also local Morans A. Of course, if you have imaging, we can also use a deep learning based approach like star-based and cell pulse and deep cell. So so using Xiaojie's method, then in the end we will achieve single cell resolution spatial transcriptomics. I think this is for the very first time, and we have been able to use this to do a lot of analysis. For example, we are able to perform single cell segmentation of the mouse heart and resolve the specially resolved gene regulatory network for this system. But another thing which I think is even more powerful is that we are able to achieve a 3D reconstruction of a whole embryo using spatial. For example, if we have the continuous slashing of an entire embryo, we are able to use we developed a series of tools that enables user slashes, and then to create the 3D point cloud, and then we can create a 3D motions. And we can also develop a deep learning approach that enable us to learn continuous gene expression pattern in the 3D space, and then with this you can perform continuous slashing at a different angle. We can also, of course, we create the 3D motions for each of the organ, and we can then quantify the names, surface area, volume of each organ, so that we can start to talk about the morphometric changes over 10 and over space of different organs. And we also develop a tool that allows us, like an online tool that allows us to interactively interact with the 3D data. And of course, once we obtain the 3D models of each of the organ, we can then start to talk about some characteristic changes during the organogenesis, including the organ expansion, shrinkage, migration, convergence, and using spatial, we are able to, for example, reveal that central nervous system is migrating during this process, and where the mid-guard start from two pitches will converge into a mature one. So since now we are able to characterize the migration pattern of each of the organ, and natural next question is that how can we predict the cell migration in a 3D space, and also predict the genes that are responsible for each of these migration. So for this, we then try to build a method that in the end we will learn the morphometric vector field. In many ways, this is similar to what we did for the ion velocity vector field learning. So instead of learning a mapping from the gene expression to the velocity, here we will learn a mapping from the spatial coordinates x to the movement, or maybe the discipline vector of the spatial movement. So the this vector is created by enabling the cells from two different time points. So like here, the red cells are from early time point of the mid-guard, while the blue cells are from a later population of cells. And then based on that gene expression similarity and also the spatial locality, we are able to map the cells from the early time point to a later time point in the physical space. And then this enablement will give us a discipline vector V. And then we can take these two for each individual cell to learn a morphometric vector field, similar to the ion velocity vector field. And of course, once we have this morphometric vector field, we are able to start to predict the migration trajectory of each individual cell. I even create an animation to show you how these two pieces of the mid-guard will start from two pieces and eventually converge into a uniform one in the end. And they also, if you look carefully, you will realize that the cells near the boundary of these two pitches actually have a strong twistiness. This can be quantified with the torsion, which essentially tells you how 3D object, the degree of the twistiness of a 3D object. And we can then perform differential analysis to associate genes that potentially related to this twistiness. And in this particular case, we revealed the OTP as a potential gene that may be correlated with the torsion. We are currently doing the experiments to validate our predictions. So as you can see, by leveraging dynamic and predictive modeling for the spatial data, we are also able to gain mechanistic insights of the self-rate migration and also changes. So maybe what I'm going to take maybe 1 more minute to just tell you that now as I mentioned, we are able to use ion velocity vector field to learn the vector field in the gene expression space, and at the same time, we are able to learn the morphometric vector field in the space, in the physical space. But these two spaces are separate, right? We didn't learn the spatial temporal models. So my current project tries to combine the gene expression changes and the physical changes into a uniform framework. We can use I would like to use this digital formation as an example to explain what I mean by learn spatial temporal models. We know during the Mammalian digital formation, we will start from like 2 digits, 1 digit to eventually form 5 digits. And if we model this system in like a 1-dimensional space, what do we and if we look at like the 3 genes that are responsible for this lineage, what we will see is that over time, this is 3 genes across the space will form like a particular pattern, and then the goal of our modeling is to take these measurements shown in this animation to actually recover the underlying model. In this case, because we are talking about the spatial temporal models, so we are not able to use ODE because that's not able to deal with the partial derivative. So in this case, we will try to actually learn PD. So here I'm going to quickly show you some. We're going to skip all this just to tell you that now using like physics informed Fourier neural operator, we adapt this method. We are able to in fact very accurately learn the this PD model directly from the data. So what I'm showing on the left is the gene expression is the gene expression for the initial state. So x-axis is the space, y-axis is the time, and the reason you see these strips is because I duplicated this initial state across this entire temporal domain. Because in this features informed neural net operate, Fourier neural net operate, we have to the input and the output need to have the same dimension. And the middle is a prediction, and the red is the ground truth. So you can see, indeed, we are able to very accurately, although there's still some noise, but we are able to learn this spatial temporal kinetics, our time and our space quite accurately. So, okay, well, I think probably for the sake of the time, I'm gonna just stop here, and I would like to greatly thank Jonathan for offering me the freedom and the support for me to work on so many exciting projects. I would also like to thank my collaborators, especially Jianhua and Yang Zhang for the collaboration on the Dynamo work, and Yang is also the co-host also on that paper. And of course, I want to thank many other collaborators from BGA, BI abroad, and of course my funding resource. Yeah. So I'm gonna stop now. So feel free to ask me any questions.
Thank you so much for the great presentation. Yeah, this was fantastic. So if you have questions, you can type them in the chat. I can also mute you if you prefer to, just to talk. And so, yeah, don't be shy. And we still have a few minutes.
Okay, maybe I can ask a quick question, just give other people's time to think about if they have any question. So right before your talk, I pronounced your tools like dynamo. Seems like after I listen to your talk, it seems like it's pronounced like dynamo. So I was just wondering, like why did you name your software as dynamo?
It's because dynamics is related to dynamics, right? What I was doing for that project is trying to learn the dynamical vector field of the system. So it's just find the dynamo is a good way to to name it.
Okay.
Yeah, no specific reason. Yeah.
Got it. So you also like present your unpublished job like Denote. I was just wondering, like, since you talk like Denote actually overcome some shortcomings of the Dynamo. So I was just wondering, like, we have not used Dynamo yet. So should we directly use Denote?
Denote currently, I think it's hidden inside the Dynamo, but it's not. It's gonna be a little bit difficult to use. Probably you need to wait for maybe a few weeks. We will release pretty soon.
Okay, great.
Yeah,
thanks.
Daniel, do you want to go next?
Sure. Thank you for the talk. It was really interesting. I have wondering, have you tested what is the performance of the perturbation simulation using not temporal single-cell RNA-seq data, but having just one data set?
Yeah, we did. What we did is we compared the result from a Potope-seq data set. Basically, we use Potope-seq data as the ground truth, and we run the Dynamo for in silico perturbation, and we just calculate the Pearson correlation of the average of the cell from our predicted from the gene expression after the lockout, and also from the prediction. We find that in general, for genes that have strong perturbation effects, our prediction is very accurate. But if a gene has relatively small changes to other genes, our prediction is not very accurate. This also, of course, makes sense because if your perturbation effect to other genes is not strong, this may be subject to the noise. Yeah.
So in that case, Dynamo will return the gene expression profile of the perturbed cell, right?
It will give you essentially a gene per cell matrix.
Okay.
For each, yeah, it's for essentially it's like the data of the gene expression changes.
Okay, thank you so much.
Okay. Any more questions?
Well, looks like we don't have any more questions. I mean, thanks a lot again. And, you are really brave to present without, the correct slides. I'm really impressed that, you were able to just do this on the fly.
Thanks, Luke. I apologize because my slide is gone and
not your fault. Yeah. I mean, thanks for not canceling. Yeah. Okay. Yeah, maybe if there are no other things, we can end here. Liang, what do you think?
Yeah, I think that sounds good. Yeah.
All right. Thanks, everyone.
All right. Thanks, Luca, again. Thanks, everyone.
Thank
Yeah.
you. Have a blessed
Bye.
day. Bye.
Bye.